It's my story on Kawasaki's disease. So a few years back, I was shadowing a doctor in the emergency department at the Woodland Children's Hospital, and I kept hearing a disease pop up. I didn't know what was going on really, but I kept hearing this disease, Kawasaki's disease. It just kept coming up day after day. Little children, 5, 7 years old, would come into the ER. They'd be rushed in with a fever, strawberry tongue, they would call it, rash all over their body, and I would hear the same routine explanation that the doctor would give. It would be, sorry ma'am, there is no other treatment. And so young me was like, I mean, there are lots of diseases in the world that have no treatment. There's lots of them, but Kawasaki's was a disease that I found to be particularly interesting because it deals with immunology. And Kawasaki's is an autoimmune disease in the sense that just like an allergy or when your body fights off disease or microbes or pathogen, your body sends white blood cells to fight this disease. However, Kawasaki's is an autoimmune disease where your white blood cells are too oversensitive, hypersensitive, and start attacking your own cells. It's like an allergy to yourself. So Kawasaki's disease, much like how when you get a cut, you get some scar tissue, you get some platelets come and just patch it all up. The same action goes on in the coronary arteries, the arteries that provide blood to the heart. But in Kawasaki's disease, these fibrin cascades or the scar tissue or the cytokine storm that begins blocking the passageway to the blood supply for the heart eventually could lead to fatal problems. These bulges are called aneurysms, and if they burst, blood would pool in the heart. They could stop blood from flowing into the heart muscles, and it would lead to a myocardial infarction, a heart attack, and it could lead to death. And so after this, I began my obsession with Kawasaki's disease. Since then I've read papers one after the other on just Kawasaki's. But the problem I found was that the scientific community knew very little on Kawasaki's. This 20-page article was literally everything they had on Kawasaki's. There was maybe a few things on speculation, a few things on treatment, but nobody knew why it happened, nobody knew a definite treatment for it, and nobody knew really how to deal with it. So going back, there are really 2 ways to study a disease in medicine. The first, I'm sure you all know about, is called a clinical study, right? So you have a population study, let's call it children with Kawasaki's in Singapore, right? And then you have 2 groups, essentially, the control group and the treatment group. Let's call the standard treatment, which is IVIG, which is largely ineffective, but it is the standard treatment. And let's compare that to, let's say, steroids. Which treatment is better? At the end, you compare your clinical outcomes, which is like fever, your lab test results, the things that doctors care about. And then, with the magic of statistics, you find that maybe steroids is a better option. This in itself was a study I already had done for capstone research, and I found that clinical studies only really change the protocol a little bit, right? It just says, doctors, instead of using IVIG, why not try steroids? We found it to be better for fevers or etc. Right? But it doesn't really change the face of medicine, it just alters it a bit. And then we come to translational research. This is what I've been doing for Catalyst. Translational research is completely different. While clinical studies work top-down, they study what's going on top, the fevers, how the patient's doing, if they're cured or not. However, for translational research, you don't need to read all this, but essentially you're studying molecular pathways, small things in the sea of science. You're studying immunomics, phenotyping, epigenetics, all these little minuscule aspects as to how Kawasaki's works in children inside the body, down to the molecular level, to the cellular level. And so from then, I continued reading some more articles. I have all these papers on like the process of immunology under skin, inside the body, down to molecular level. And at one time, I spent $40 on a Nature article for this one specific article I needed from 1998, and it kind of helped me like put a picture as what this disease was and what people knew about it already. And so I went to, of course, the most famous Institute of Translational Immunology in Singapore. This is academia. It's by Duke-NUS Medical School, and it's a huge institute dedicated to just studying immunology from this perspective. I came to this guy. He's the primary investigator of this institute. Essentially, he's the boss of the place, right? He runs all the experiments. He runs all the projects. I went to him. He's a pretty friendly guy, and I told him I have this idea on how to treat Kawasaki's on a molecular level, and he was like, we'll see. And that essentially meant 2 months of exploratory work. Here's some photos from how it looks like in the lab. keep all these in cryogenic tubes. All the work was done under fume hoods. And I spent months just looking at what they were doing, and another month spending learning basic procedures, basic experimental techniques and they had a pretty nice view too, but it's pretty nice. This is my usual setup, right? This was like for our RNA extraction thing I was practicing. This one would maybe be analyzing the phenotyping. We would count cells, we would be putting it in machines, just practicing my basic lab skills. And then finally, he gave me a box, right, in a little frosting, this cool place, and said, you can start. Of course, I have already given him my design of study, my experiments, etc., and I was ready to begin. So, the experiment. So how would I approach this topic? I had done all this research on basic immunology, the basic processes, and I started with essentially the basics, the natural processes. There were two of them. One of them was called autophagies, right here. It's cell hibernation. Sometimes cells, when they're in hard conditions, they want to prolong their lives, so they cut off a limb, cut off an arm through lysosomes, and they just keep on living, right? Sometimes they go into apoptosis. This is more familiar, maybe. It's when a cell commits cell suicide, complete destruction, and it completely goes away, right? So naturally, these two processes are equal. They balance out. You want to maintain an equal amount of immune cells, for example, and these two systems would keep that in check. But unnaturally, sometimes, for cancer, for autoimmune diseases, sometimes we get an imbalance. This is when cells are living too long. And this is Kawasaki's disease. Kawasaki's disease is when there are too many T cells, there are too many hypersensitive cells, and they're wreaking havoc all over the body. And so I was looking for maybe a drug, treatment X, that could possibly stop autophagy, right? Stop them from prolonging their lives, and allow apoptosis to naturally stop Kawasaki's disease. This is how I frame my method, my line of reasoning. And so what was this mystery X? This was one of the biggest challenges I found, but I mean, one of the shortest articles I came across, it's like a 2-page article, I think. It says here, hydroxychloroquine preferentially induces lysosomic degradation. And what does that even mean, right? Right here, right? But what it does is that I understood that autophagy required lysosomes, and hydroxychloroquine could be a possible method of inhibiting autophagy. Back to here again, right? You want a drug, hydroxychloroquine, HCQ I call it, that would stop autophagy. This would probably be a mechanism to possibly treat Kawasaki's disease from a different angle. I began with a trial run on Duke-NUS cells. Duke-NUS cells are not Kawasaki cells. They're immortalized primary cell lines from a 6-year-old leukemia patient, which essentially means that these are immune cells, T cells, that rapidly keep on dividing, keep on creating progeny T cells, and they naturally undergo autophagy. They naturally undergo apoptosis as they would naturally, but they're readily available, they're easy to use, and there wasn't very much risk at it. So my experimental design broke down into 3 basic steps, right? Culture these cells in growth media, allow them to grow, right? Treat them with HCQ, the hydroxychloroquine, and see if autophagy was inhibited. These first 2 steps are quite short, but they took a lot of time to master, and each step is like a lot of things to do. So these 2 steps seem fairly self-explanatory, but the 3rd step was kind of how do you measure autophagy, right? How do we measure autophagy? My solution that I came across after exploring all the mechanisms and machines that they had at the institute was through a fairly bland machine called the fluorescence activated cell sorting FACS flow cytometer. So let's break it down for you. Fluorescence. So it was some machine that used fluorescence to sort some cells, and cells flowed through a cell meter, a thing that measured cells in its fluorescence. That is what it was essentially. But inside this glorious machine was an intricate process. This is maybe the best diagram that can kind of show you this. But what it does is it sorts cells and counts every single cell, every single one of them, like millions of cells in their size, their granularity, which is like the texture of a cell kind of, and the fluorescence. These 3 things were key for me to discovering how to measure autophagy in cells. First is the physical properties, size and texture. How this worked was that when each cell was funneled through a tube, one at a time, through a laser beam fired, the diffraction and refraction patterns from this cell would create a shadow, and you can see the cell got bigger, it would be a bigger shadow. So essentially, it was a neat way to measure how big a cell was. And then the scatter around here, we see, was the granularity, right? When light passes through all of the internal structures, how it scatters, and if that was measured, that would measure the texture of the cell. And so if you just plot it forward scatter by side scatter, this is an example histogram, dot plot, you would get like a grouping of cells that would be similar. And you could eliminate the outliers, the dead cells, right? The ones that are too small, have weird textures, right? And you can find these cells and say these cells are the same. They're the same type of cell. Next mechanism was fluorescence, right? So you pass cells through a laser, and if they had some fluorescent properties, it would fire off, and these indicators would measure the fluorescence, right? This is, again, an example histogram of measuring fluorescence intensity by cell count. And so you get all these different cells with different fluorescence measures, etc. So this is all well and good, but how did it apply to autophagy? The neat thing was the process of autophagy, while very neat, very complex, was that it required surface antigens for it to form the autophagosomes, to allow it to keep on living. There were these specific autophagy markers, and we could use manufactured monoclonal antibodies to specifically tag on to these surface antigens and tag them with fluorescence. We could literally tag every single cell who presented this specific antigen with a tag, a fluorescence tag of fluorophore, I believe it's called. Here is an animation of how it worked. Each cell would all be measured at one time. This took quite a long time. I think it was about like 24 hours for an entire sample to run through this machine. And so we're back to this, right? Now we have our answer. Culture them, treat them, and then use FACS flow cytometry to measure if autophagy was inhibited. This is my sample data. This is one sample I used. It measured forward scatter, which was the size scatter, which is the granularity, and I can say that these cells can be ignored. These are dead cells. These haven't grown, and these cells, I just grouped them off. These cells are ones that are of interest. These are cells that are big, are same type, and can be studied. With these cells, I found each of their fluorescence, and I could measure that 530 cells in this sample were in autophagy. That's how this is what I did for every single sample I had. So here's my sample data, right? I had 10 samples. I used a dummy variable, 1 or 0, binary, indicating 1 for I used HCQ, 0 for no HCQ, and I would measure autophagy. So if you just take a quick glance at this data, right, just cutting off here, it kind of looks like if you treat HCQ, the number of autophagy cells decrease, which is what we want, right? But how do we quantify that? Like, how do we quantify this data? It's this data, and here is when a demonstration in R would come in handy. This is the magic stats. And R is a programming language that I've also grown to learn with this project, and I will show you a demonstration. Oops. So this is a developing kind of software thing, and so first I wrote this script a while ago. It's an algorithm and a script, and the first two things just finds the mean, the average, right? Let's run this. A view data, right? Same exact table, but now it's in a programming interface. And now we find the mean. 483.8 for those with HCQ that was 1. It kind of makes sense. The syntax doesn't matter. If we tried with 0, it would be 707, right? So at first it looks like it kind of worked, right? 483 is lower than 707. We can say that autophagy was inhibited. However, we can't stop there. I mean, it could be entirely random. That was the issue. And so I created a script that kind of modified a statistical test in which we modify a type 1 error, a type 2 error to ensure that my results were not random. So the next part is a series of code that, and this is for a loop for those who are interested. And I run 100,000 simulations on this specific data. And I would end up with a value, 0.05674. What does this mean? This value is called a p-value, right? And essentially it meant if we assume that the HCQ had no effect, there is 5.67% chance that our data can be received. Essentially it means that we have 95% confidence with a type 1, type 2 error, that there is statistical significant results of our data. So we can confidently say that there was a difference. 483, I believe, and 707 is significantly different. And so now we're back to R. And so the experiment worked. Yay. So we found that HCQ did in fact decrease autophagy. But if you remember back to the slides, that wasn't it. That wasn't the entire process. We have to allow for apoptosis to eventually kill off the rest of the excess T cells. And that is my future steps. So for the past few months, I've been spending maybe every other day skipping all my 80 classes to go to this lab to do this work. And I've been up to this, right? My future steps would require me to, instead of using Jurkat cells, use Kawasaki PBMC, which are Kawasaki T cells, but these are hard to get, they're hard to find, and they're hard to store in the refrigerators, and so that would come next. I'd also have to measure apoptosis and confirm that hydroxychloroquine did in fact decrease the total amount of T cells in Kawasaki patients. And so hopefully, after maybe a few more weeks, I'd be able to confidently say that hydroxychloroquine, a new drug that nobody has really tried before on the disease, does in fact decrease Kawasaki's and helps patients live longer lives. Thank you.